So now we are starting microbiology, the clinical bacteriology. So today in this video we'll talk about the gram-positive laboratory algorithm. So how do we identify the various gram-positive bacteria that includes bacilli, cocci and branching filament. I will discuss one by one in detail. So as you have already know about the various strains of the bacteria. So one of the strains of the bacteria that helps to identify the various bacilli easily is the gram strain. So just to revise, the gram positive bacteria appears the purple or blue color, whereas the gram negative bacteria appear red or pink color. So saying that the gram positive bacteria after the gram stain appear purple or blue color. So three types of bacteria can be identified in the gram positive bacteria. That could be the bacilli, that could be the cocci, that could be the branching filaments. So various branching filaments bacteria are again divided into two types. So I am going to describe first the branching filament, then I will discuss about the bacilli, then at the last I will discuss about the cocci. So the branching filament in the gram positive bacteria are again divided into aerobic and anaerobic. So you can say that whenever you see in the microscope after the gram stain, you see the bacteria that have branching patterns. So you see the bacteria that have branching pattern like this. So you see the bacteria after the gram stain that has purple or blue color and it have branching filament. So you immediately know that it is branching filament gram positive bacteria. And there could be the two types in the branching filament. There could be the aerobic branching filament gram positive bacteria, and there could be the anaerobic branching filament gram positive bacteria. The aerobic branching filament gram positive bacteria are Nocardia species, and the anaerobic gram positive branching filament bacteria are Actinomyces. So these are the examples of the branching filaments. Now so another type of bacteria you see the rod type of bacteria. So after the gram stain and on the microscope you see the rod like structure like this. You see the rod like structure. So after seeing the rod like structure, so you immediately identify the structure is bacilli. So it is purple color and you see the rod like structure and you identify it as the gram positive bacilli. The gram positive bacilli again can be divided into two types. That could be aerobic and that could be the anaerobic. The anaerobic gram positive bacilli are the Clostridium species, whereas the aerobic gram positive bacilli are Listeria bacillus and Corynebacterium. Again, the anaerobic gram positive bacilli are the Clostridium species of the bacilli, and the aerobic gram positive bacilli are Listeria bacillus and Corynebacterium. So these are the examples of the aerobic and anaerobic gram positive bacilli. Now let us discuss about the cocci. So in the microscope you see the pinpoint structure like this. You see the pinpoint purple or blue color structure, blue color bacteria. So that are the structure of the cocci. So after the gram stain you see purple color dot like structure and you identify it as gram positive cocci. So those cocci could be in chains and could be clumped. So two types of presentation of the cocci you can find. So they can be the clump like structure like this, or they can be on the chain. So they can be on the chain structure. So the chain structure of the cocci is the Streptococci, and the clump structure, that grape like structure of the cocci, is Staphylococci. So the cocci is also divided into two types, that based on the catalase. One is catalase positive and another is catalase negative. So the catalase positive cocci are staphylococci. I have already said that the staphylococci are clumped structure and appears like grape-like structure, clumped grape-like structure. The staphylococci can again be divided into coagulase positive and coagulate negative. The coagulase positive cocci, staphylococci, is Staphylococcus aureus, but the coagulase negative cocci is again uh, divided into or categorized into novobiocin sensitivity. So novobiocin sensitive coagulase negative staphylococci is Staphylococcus saprophyticus, and novobiocin sensitive positive coagulase negative staphylococci is Staphylococcus epidermidis. So do not confuse it is. So on the staphylococci, coagulase positive staphylococci is Staphylococcus aureus. The coagulase negative staphylococci is again divided on the basis of novobiocin sensitivity. The novobiocin sensitive coagulase negative staphylococci is Staphylococcus epidermidis, and novobiocin unsensitive coagulase negative staphylococci is Staphylococcus saprophyticus. So this is about the staphylococci. Now let us move towards the streptococci. So as you already know, the streptococci is catalase negative gram positive cocci that occurs in chains. So it is divided into three types on the basis of hemolysis. So it is alpha hemolysis, beta hemolysis, and gamma hemolysis. Alpha hemolysis, beta hemolysis, and gamma hemolysis. On the alpha hemolysis, it is partial hemolysis and occurs green, whereas in the beta hemolysis, it is complete hemolysis and it is clear. And in the gamma hemolysis, there is no hemolysis, there is no actual hemolysis in the gamma hemolysis, and it grows in the bile. First of all, let us talk about the alpha hemolysis streptococci. So the alpha hemolysis streptococci is again divided on the basis of optocin sensitivity and bile solubility. So optocin sensitive and bile soluble streptococci are Streptococcus pneumoniae, which is encapsulated. But optocin sensitive and bile soluble, the optocin insensitive and bile insoluble group of alpha hemolytic streptococci are viridans group of streptococci. They are Streptococcus mutans and Streptococcus mitis. So again I am going to tell you, the streptococci is divided into alpha, beta, and gamma hemolysis. On the basis of alpha hemolysis, beta, and gamma hemolysis, the alpha hemolytic streptococci is divided again on the basis of optocin sensitivity and bile solubility. The streptococcal bacteria which are optocin sensitive and bile soluble are Streptococcus pneumoniae, which is encapsulated, whereas optocin insensitive and bile insoluble streptococci are viridans group of streptococci. Those are Streptococcus mutans and Streptococcus mitis. The beta hemolytic bacteria are again divided into bacitracin sensitivity and PYR status. The PYR status denotes the property of the streptococci to enzymatically hydrolyze the L pyrolydyl beta naphthylamide, that is PYR. So bacitracin sensitive and PYR status positive patient are Streptococcus pyogenes. Those are the group A streptococci, whereas bacitracin insensitive and PYR negative status are group B streptococcus. Those are Streptococcus agalacticae. And again, the streptococci are divided on the basis of gamma hemolysis. So those are again divided into growth in 6.5% sodium chloride and the basis of PYR status. So the streptococci the streptococci of which the growth occur in 6.5% NaCl and PYR status positive are Enterococcus. Those are Enterococcus faecium and Enterococcus faecalis. And those streptococci which do not grow on the 6.5% NaCl and PYR status is negative are non-Enterococcus bacteria. Those are Streptococcus bovis. So these are the various examples of the gram-positive bacteria. Those are bacilli, cocci, and branching filament that have been described in detail. So before actually starting the microbiology clinical bacteriology, I want you to know about the gram-positive laboratory organism. I mean gram-positive laboratory algorithm of the gram-positive bacteria. I hope you understood this video. Thank you.